Okay, thank you for the introduction. Thank you for the opportunity for me to talk in this educational session. Okay, so to begin with, I'd like to start with thank you. I have taken some slides from my colleague Katie Chen and colleagues Nanyin Zhang. They're very kind to share some of the materials that I use for this lecture. So this is about introduction to functional connectivity. So I would like to digest the term a little bit. So what do we mean by functional connectivity? I think there's by functional, actually, we mean that the data we are talking about is about activity. It's about the dynamics. It's also about the emergent patterns from the brain networks or circuits. By functional, we also mean that whatever pattern we are trying to make sense of and has to be related to brain behavior, cognition, perception, action, so on and so forth. By connectivity, our goal is to actually talk about the network in the brain, and that means the pattern of interaction between regions, between voxels, between populations, between neurons. And uniquely is using the activity to make inference about the connectivity. So let's say we have the brain, there are neurons, there are regions, there are voxels. Let's say simplify that case into just 5 different regions, and they are structurally connected, and we take measurements from them, and we usually do the analysis without any pre-assumption about how they are wired to begin with. And then we just simply maybe measure the correlations between different regions or voxels. And based on that, we make an inference about the underlying connectivity. That underlying connectivity is a result of some sort of an inference. It does not have to be the structural connectivity, but sometimes these two correspond pretty well to each other. So most of the people, when we actually analyze the functional connectivity, and we use the resting state. In resting state, we know that the brain does not rest at the rest. The activity is pretty intensive and very complex. And this is displayed at a much faster rate than it occurs. So you can, even with this raw data, you can visually appreciate lots of interesting patterns that's going on and off. And sometimes you can see the whole brain goes blue, and sometimes the whole brain goes yellow. And to look at the second order structure of this data, you can use the correlation. In this case, we use the tool of fMRI, and instant call is a very convenient tool to explore the data. And you can choose a voxel of interest, any voxel that you like to explore, and you look at the signal from that voxel and correlate that signal with the signals from elsewhere. And then you can look at the different patterns. And this second order structure of the raw activity patterns seems to be very well structured. And that's what people usually refer to as a functional connectivity as observed with resting state fMRI. This is started with the seminal work from Biswar and over 20 years ago, and discovered that the spontaneous activity correlates, showing a pattern that looks very similar to the task-evoked activity pattern. And depending on what is your location of interest, you can look at a number of different networks. And there's about a dozen of them. Looks very robust, very consistent across subjects. There are termed with some names, and those names actually it's not always clear where the names come from. But if you look at the patterns, for example, these are the patterns extracted from spatially independent component analysis. And they generate some pattern looks fairly similar to seed-based correlation. You compare them to the patterns that one would observe during some sort of a task, and then you get some sort of these functional terms emerge with it. So in fact, that we see this visual network, sensory motor network, they do not exactly mean anything except that they overlap with the sensory motor cortex, corresponding cortical areas, and they may be corresponding to the same type of patterns that will be activated by those corresponding tasks. And the functional connectivity can be evaluated with different spatial specificity. And according to the David Van Essen's talk just now, and there's a dense connectivity patterns, basically describes how every voxel is interacting with every other voxel. You can also look at these regions. Then the question is how we define the pulses. You can also look at the network-to-network interaction. In this case, very often people use spatial ICA to actually extract those components and look at the component-to-component interactions. This pattern looks sometimes very, very consistent across different sessions within the same subject. So for example, this one shows that across many different sessions, there's extremely high level of overlap. This does not hold true for all the networks, but for those reliable, about a dozen networks, this is quite consistent. And this also makes it very interesting to look at another data set, which is complementary to Human Connectome project. It's named as MyConnectome, and it's released by Stanford Group. It look at a single brain and scan it repeatedly for over a year, which is an extreme case for those who are interested in looking at reproducibility of different network patterns. To make this useful, and one may think about if this network looks so consistent within an individual, whether or not someone can use it as some sort of fingerprint of everyone's brain. And then this is the work done by a group from Yale and Emily Fins, and it shows that you can look at parcel-to-parcel correlation matrix for a number of subjects. And then for a specific subject, you can simply do a correlation and you find out the one that matches the best. That turns out to work pretty well, even though this analysis is very, very simple, but it turns out to work very well. And it works well, especially when the data are acquired for a relatively sustained amount of time, and then so you get a reliable functional connectivity matrix to begin with. And what's also interesting is that if you do this type of subject identification task, and if you use the resting state data alone, and you can do pretty well, but if you combine the resting and the task data together, you do it much better too. To further move on from this, I think that it will be useful to make predictive models. The predictive models goes beyond this subject identification, but target towards maybe disease progression or prediction of individuals' behavior or symptoms. We also know that the resting state data, even though it has rich information, it tends to be noisy and hard to characterize or understand sometimes. It contains lots of different noise, signals, and artifacts. So this dates back to again, about 10 years ago, and many people studied this, including the results showing that the respiration, the breathing patterns, or the respiration volume per time turned out to correlate very broadly with the fMRI signals from the brain. So if we buy the argument that this signal is of no interest to the brain, and then this turns out to be physiological noise or artifacts. And then if we do the regression analysis to look at a different type of patterns and try to see that the whole brain activity can be explained by noise or different types of noise or artifact versus a signal, arguably there's about maybe 20% of them are signals. All the others might not be that useful. And this argument has triggered a lot of intensive studies on the physiological noise correction. And another big debate is whether or not we should regress our global signals. And for over 10 years, I still don't think that people have settled on this question yet, because it really makes a big difference. If one regress out global signal, you see some of this, especially these anti-correlation patterns. If you do not, you may or may not see those patterns. But you get a much less, sometimes much less, specific patterns of functional connectivity. Many people continue to investigate on that. The reason why we want to regress it out, perhaps it has a good reason, Because if you look at, this is work by Carrie Shimali, and showing that if you look at the heart rate signal, and the heart rate signal goes up and down, that signal turned out to correlate so broadly with everywhere in the brain. And so this might be suggesting that the global signal has a non-neuronal physiological origin. So for this reason, it might make a whole lot of sense. However, other people also find that if you take the neural signals from a single location in the brain, and you correlate that signal with the signals from elsewhere in the brain, and you end up with a very broad global correlational patterns. And the neuronal underpinning of that correlational pattern turned out to be very well structured, showing a very broad gamma band or high gamma band activity. So it may have a neural origin. So what do we do about this? One way I think is a good way, and is a conservative way, in my opinion, is to look at the bold versus non-bold component. This is a work by the Prantik Kantu from NIH, showing that if you take fMRI signals from multiple echo times, and then you do the multi-echo ICA, and look at individual components to see how the activity patterns vary or depends on the echo time. And you see that relative signal changes, actually, if the relative signal changes increase linearly with echo time, that seems to suggest this has a bold origin. Otherwise, if the signal goes pretty flat as a function of echo time, that suggests it may be a non-bold component. If you sort of quantify this, the degree of being bold with some value, and you plot it out, you end up with this sort of like a L-shaped curve. Take a threshold, you maybe separate out some of the bold component versus non-bold component. Even though we are not 100% sure that all the bold components are useful or neuronal, however, this is conservative ways of teasing out some of the obviously superior noise components or artifact components. And then that could serve for the purpose of cleaning up the data somewhat. The next topic is that when we look at the correlations between voxels, regions, or networks, this is by no means limited to the resting state, even though that resting state is mostly used for this type of analysis. So here, for example, if someone watched a movie and watched the movie twice, and you end up with that the activity looks so consistent between the two times. For example, this shows like two example voxels, one from the V1, the other from V4. You could look at the signals that you acquire from session 1 versus session 2. It looks really, really close. And this type of task, unlike the resting state, seems to engage the optical radiation, make them actually correlate better with one another, and much more than otherwise the spontaneous activity would do. So this makes it very intriguing. Just like the resting state, and when someone watches a movie, the data looks so irregular, statistically speaking, but it offers the same opportunity for us to look at how regions are interacting with one another and how do we make sense of it. The real power comes always this. So if the signals look so consistent across different sessions, we probably can take a voxel time series from one session and correlate it with voxel time series from other session. So for example, if we look at functional connectivity in the same way as we do with the resting state, and we can say we choose a seed voxel, in this case is a region number 1, and we look at the region number 2, or other regions, we look at their correlation. The correlation contains the signal generated by the natural stimuli. It contains the signal generated due to the spontaneous activity or fluctuation. It also contains the contribution from all kinds of noise. But now if you either have the same subject to watch the movie or receive the stimuli, the natural stimuli for the second time, or look at two individuals who watch the same stimuli, and you look at the activity correlation between them. Now you look at the same voxel and you find out that from the first subject or for the first session, you have a signal S1, and you have a signal S1 again, which is the repeat signal. This repetition is not perfect, but when you look at the correlation between the signals from the same voxel, but between different sessions, you can look at, as shown in the previous slide, that you can end up with a very high correlation. This reproducibility may tell you the areas that are being activated by the natural stimuli. That activation might be a little bit underestimated. However, it's reliable enough to show interesting patterns. To do this in somewhat related but different way, if you take a voxel or region from one brain during one session, and you correlate that signal with everywhere, including itself, from a different session, either from a different individual or from the same individual. In this case, if you look at the signal, you have an ACE1, it's a signal evoked by the natural stimuli. You have the spontaneous activity or intrinsic activity, I1. And you look at the other session, you're going to have these three terms, and you look at the cross-correlation between them, you only see the contribution from the signal actually evoked by the task itself or the stimuli, instead of contribution from either the noise or the spontaneous activity. In my view, I think this is very powerful, this is very useful, even though the reproducibility may not be perfect, so we have to keep in mind that this type of intersession or inter-subject functional connectivity might be an underestimation of the real functional connectivity evoked by the task. This can be useful and allow us to interpret the pattern of functional connectivity in different ways. Here shows an example. If you do this type of intersession or inter-subject functional connectivity for a resting state, because of the lack of the task, you're going to end up with nothing, which is very intuitive. If you look at if you let the subject listen to a narrative story, but the story consists of the words that are all scrambled, you hear the words, however, and you do this type of inter-subject functional connectivity, you're not going to see any pattern. Now, if you scramble not the words, but the paragraph, you do see the default mode network actually this time emerge. If you look at the intact story, and then the default mode network looks more pronounced. So now we see the default mode network, and this looks the same default mode network as we will see during a resting state. But now we have an interpretation for this type of network pattern. In this particular case, the default mode network pattern seems to indicate the amount of cognitive loading for this natural language comprehension task. Lastly, we often look at the whole brain scale, but that's not necessarily the only option. We can look at the system level, we can look at the laminar level, we can even look at the signals within a voxel. What do I mean by that? For example, if you look at only the visual cortex, in this case, you look at the resting state, and you pay attention to the fine-grained pattern in the resting state, you can see that this seems to be a very interesting pattern that very often people would not pay much attention when they look at the whole brain scale. Now, if you look at this, present the subject a movie and do some type of intersession or inter-subject functional connectivity, you can see this pattern somewhat looks similar, but there are some differences as well. And both of these two patterns, if you compare with the same type of patterns that one would see during doing a retinotopic mapping experiment with eccentricity and the polar angle modulated stimuli, you will find out that none of these patterns look similar to the organization of the polar angle and somewhat similar to the organization of the eccentricity. However, they're not the same, especially they don't show up this anti-correlation between the fovea and the peripheral regions. We can also look at apply the independent component analysis to the network, to the activities within a specific system, in this case, still the visual cortex. And you can end up with a number of different ICA components. And we may interpret this as the subvisual networks, or maybe not. But nevertheless, you see some of the bilateral patterns, some of the unilateral patterns. You can look at the activity, the component-to-component correlation, and on the basis of that, you can end up with some puzzles or clusters, and you find out that the foveal early visual cortex, peripheral visual cortex, dorsal and the ventral stream extrastriatal cortex seem to show up as different clusters. And then you can look at this type, you can use this one to postulate not the whole brain, but only the visual cortex. This end up with something closest to the HUCP 2016 Nature paper postulation, but looks quite different from many other postulations. However, it still looks different from that Nature postulation. One more argument we like to make is that if you move a seed location to an extrastriatal cortex, in this case, fusiform face area, you look at cross-section functional connectivity, you can end up with a pattern that looks just like a face selective network. And even though the analysis is targeted to this network, we can actually go back to the movie that presented to the subject. You can look at the different, the network activity goes up and down. You can find out that when it goes to the peak, it corresponding to the time points where the movie contains the human faces. So this type of stimuli actually give you more options for you to make to interpret the patterns of functional connectivity. Another one, if you push down the resolution to look at the lamina profile of the functional connectivity, we know for years there's a canonical microcircuits. This microcircuit shows the forward and the back connections that end up with different the cortical layers. So the extrinsic connectivity has this lamina profile. Now, this lamina profile has recently been shown, not 2 years ago, it's been shown by Huber et al. From NIH, showing that if you look at some seed, for example, ACE1, which gives input to the M1, you show that they end up with a profile of functional connectivity observed with the CBV contrast fMRI. You can see that this actually peak roughly at this location, and this corresponds pretty well to this canonical circuit pattern. Now, if you look at a premotor cortex, if the premotor cortex sends back the backward connections to the M1, you can see that there's like two peaks, like two hills, and this actually looks pretty similar to this to the superficial and the deep layer for the backward connection. I think this is it's very interesting, and there needs to be more work to be done along this area. Now, if we give a further push, let's say we look at a single voxel, but in this case, if you look at single voxel, there's only one single signal if you look at a voxel with fMRI. But if you put a probe into there and you pick up different neurons using single unit activity, you will end up with seeing there are different type of subpopulation of cells even within this very small single voxel. And you pick up the activity fluctuation from each one of these subpopulations, and you correlate with the fMRI signals across everywhere else in the brain, you'll find out the very different patterns of correlation. And this pattern suggests that the correlation to a single voxel, depending on which subgroups of neurons we are talking about, can vary substantially. Lastly, to look at that, according to the last slide, and we also know that to understand what the pattern of functional connectivity we are looking at, we sometimes should go to the animals instead of look at the humans alone. In the animals, and there are less of the logistic, practical, and ethical hurdles, and many of the networks that we see in the humans and have some analogous networks in the animals. Very known one is the deformal network, which has been found in macaque, rat, and marmoset among many different species. And this patterns, using this homologous or analogous networks in the animals, we can have more targeted analysis and study of what of these networks to see what they do. There's a barrier, there's a major difference between looking at the brain in animals versus the humans. Animals, fMRI usually are anesthetized, but there are more and more groups are doing this on awake animals. Some of them are using, this is the work from Nanyin Zhang and Penn State. Well, what they did is that they trained animal to get used to this scanner environment, and after many days, You can see that the stress level goes down as you see the heart rate and the respiration tend to stabilize in a low level. And then you can also end up with a lower motion, and you can then prepare the animal, initially briefly anesthetize the animal, put them in the scanner, let them wake up, and then run your study when they are fully awake. And doing that is not only for the rat. Actually, people have been doing this for a number of different animals, and so this is a productive line of research. And this is, again, the data from Nanyin Zhang, and it shows that if you do parcellate the rat brain in a roughly similar way as how people parcellate the human brain on the basis of the functional connectivity profile throughout the whole brain, you can end up with different parcellations, which he refers to as the functional atlas of the rat brain. Doing functional connectivity in the rat brain is not just a simple backward translation. Many of the things we can do in animals, not easy to do in humans. One of such things is the optogenetics, and this is an old study showing that if you perturb the circuit, by the way, either by excite the circuit or inhibit the circuit, you can observe the network response of it. So, given the interest of time, I'd like to wrap up. And most of the people doing the resting state functional connectivity is easy to acquire and easy to analyze. Now, given the large-scale efforts of HCP and others, and we no longer need to acquire a lot of the data. We often can just analyze data mining of the existing data. However, the interpretation is not always easy. And we know the resting state spontaneous fMRI signals are noisy and there are artifacts. Some of them have been increasingly taken care of, and some of them remain of concern as a broad clinical application. However, I would argue that there are another opportunity of looking at the activity, which is a task-evoking activity during some naturalistic stimuli. Those signals, like the spontaneous signals, that are also irregular and statistically of interest, but it's well under control. It's more behaviorally relevant and less susceptible to the noise, and it's at least a different type of measurements complementary to what we see with spontaneous activity. There's a tremendous need for multi-scale functional connectivity, and the whole brain analysis has been a focus, but that does not have to be the only focus. And then you really need to be a strong push push the resolution down to the level of cortical columns and the lamina, and then breach different scales. I think it's still within grasp of our lifetime. And the other thing is that to look at the animals really helps, and we have the need to understand the origin of the connectivity patterns we see with fMRI, and we need to go from correlation to causality. With that, thank you for your attention. I'd like to, we'll be happy to answer any questions if there are any.